Welcome back. So the first topic for the second session is about the single domain antibody discoveries using different platform technologies. So comparing to the traditional monoclonal IgG or the IgG fragment like a Fab or single chain FVs, single domain antibody comes with a much smaller size and better tissue accessibilities. And the single chain format gives it a unique flexibilities in engineering to fit different kinds of formats. And comparing to the single chain FVs, the single domain or the VHH is generated in vivo by animal immunizations. So it has a pretty decent developabilities and manufacturabilities. And most of all, the single domain antibodies has been validated in terms of the efficacies and the safeties by a lot of clinical pipelines. Over the last decade, the single domain antibody is widely used in bispecific or multi-specific antibodies as a building block, or in cell therapies like CAR Ts. In recent years, the single domain antibodies is gaining more and more interest in gene therapies, especially in the in vivo targeted delivery for the virus or for the mRNAs. So this is a summary of some high-profile single-domain antibody-based therapies. So far, there are 4 approved single-domain antibody-based therapeutic antibodies for the indications like oncologies and autoimmune diseases. There are more than 140 pipelines globally. Most of them are in the preclinical or early clinical stage. ProBio offers 3 different approaches to generate single-domain antibodies. Both the immunized and the naive alpaca libraries are built based on the phage display technologies. Alternatively, we can also screening for single-domain antibodies at single B-cell level using the Beacon platforms. We will share more detailed technical information in the next couple of slides with these 3 different strategies. After recovering candidates, these leads will be further characterized by in vitro assays, followed by engineering such as humanization of affinity maturations and in vivo animal disease models, and also the developability assessments. It's a pretty standard process. So these 3 different platforms to generate a single domain antibodies has its unique features, and it comes with its specific application scenarios. For instance, if your top priority is really good diversity, and the timeline is also really sensitive, so people usually will go with the Beacon-based single B-cell strategies. Alternatively, the immunized phage display libraries can generate antibodies with higher affinities, but when your target antigen has a really high homology to the camelid animals, it may be potential issues because of the immune tolerance. Under that circumstances, we can resort to the naive library. Without the immunization, you can so basically you can eliminate the immune tolerance issues, and you can generate the antibody leads using the ready-to-use naive library. But one of the main issues for antibody coming out of the naive library is the affinities. Comparing to the other 2 approach, the antibody affinities is somewhere around 10 to the -7 to -9, roughly 100-fold lower or worse than these 2 immunized-based approach, for both single B-cell approach and the immunized approach. And this gives you a general idea about the workflow for each of these different strategies. For single B-cell based strategies, we start with the animal immunization. After recovering and enrich plasma B-cell from the PBMCs, these B-cells will be loaded onto the chip for single B-cell antigen binding screenings. The positive B-cell will be exported for recombinant expression and confirmation by antigen binding affinity and cell-based functional assays. And after the functional confirmation, the top candidate will be sequenced for scale-up production and in vivo validations. Alternatively, for the immunized phage display approach, we also start with immunization. The PBMC will be licensed to recover the mRNA and to build the phage libraries. Usually the library will be panned and screened by different kinds of antigens, including soluble proteins and recombinant cell lines or any other antigens of interest, followed by candidate sequencing, recombinant production, and functional validations. The naive library workflow is pretty much the same as the immunized library minus the immunization and the library construction part. So the naive library takes much less time with the sacrifice of antibody affinities. So for the single B-cell approach, there are 3 key steps. The first key step is to enrich for the plasma B-cells. When we were doing this kind of project, first we will use the antigen to enrich for the antigen positive memory B-cell. And the memory B-cell will be converted into plasma B-cell in vitro by proper stimulations. And these plasma B-cell will be loaded on chip for the screenings. The key feature of this Beacon platform is it is able to run multiple runs of antigen binding on the chip so that you can successfully narrow down your candidates from thousands of positive B cells to probably 50 to 200 positive B cells. And even with, let's say, 100 positive B cells, the traditional approach following the B cell screening is to do the sequencing, gene synthesis, plasmid preparation, and recombinant production. It's gonna be a huge amount of work to do the whole process with 100 or 200 positive B cells. So that's why we have the third key step, the ProSpeed expression platform. So instead of doing sequencing and the gene synthesis and the plasmid preparations, we will do the PCR with the positive B cell to clone out the antibody fragment, the variable region fragment, and clone it into a linear plasmid by homology recombinations. It's much convenient, much more easier than the traditional gene synthesis and the ligation and the PCR reactions. Then we can take this reaction mixture directly for transient transfection with mammalian cell lines like CHOK1 or HEC293s. It's exactly the same as a traditional circular plasmid-based recombinant production. And after this recombinant production, we will harvest the supernatant for binding and functional assays, cell-based functional assays. So with this ProSpeed expression, we're able to handle hundreds of positive B cells with a relatively low cost and a relatively shorter timeline. Besides the single B cell, the phage display also provides an alternative option for the single domain antibody discoveries, including both the naive library and the immunized library, as I mentioned in the previous slides. So in addition to the library itself, the panning strategy also plays a very crucial role in the phage display-based antibody discovery strategies. Here is a list of the most common panning strategies we often use nowadays. The liquid panning is basically an approach to capture the antibodies by biotinylated antigens in the suspensions, while the solid antigen panning is to capture the antibody by the antigen immobilized on the plates. The cell panning is basically using engineered cell or cancer cell to capture the antibodies. For any project, we will typically combine different kind of panning strategies to improve the overall success rate and to maximize the diversity of the epitope and the diversity of antibody sequence. Traditional phage vector, phage plasmids, the antibody is actually fused to the P3 proteins of the phage. So that's how the antibody is displayed on the surface of the phage. But that also brings a major issue. These phage will significantly interfere the antibody antigen binding. So that's why you often see non-specific binding background signals in ELISA or flow cytometry assays when you use the phage supernatant. So that's why when we use this phage vector, it comes with a Amber stop codons. When we induce the cell with IPTG, the expression of the antibody will stop at the Amber stop codon as a soluble version without the P3 proteins and the phage. So this soluble version of antibody candidates will greatly reduce the signal noise, the non-specific background in ELISA and the flow cytometry analysis. And also it makes it possible to run the SPR affinity ranking with the phage supernatant. So after generating the top candidates, the next step is engineering. The humanization of a single domain antibody is pretty similar to the humanization of like rodents or rabbit antibodies. It's still based on the CDR grafting and framework back mutation strategies. The only differences is there are several conserved residues in the framework 2 of a VHH. So there are certain special design for the back mutation when you consider doing the humanization of VHH single domain antibodies. Over the last decade, our success rate of a single domain antibody humanization is roughly at 91% plus 5%, so that's 96%. As I mentioned previously, if the antibody is generated from the naive library, the affinities might not be very good. So in that scenario, we may need to do affinity maturations to in vitro affinity maturation to improve the affinities. So ProBio offers a pretty unique strategy for affinity maturation. It's called two-step library-based approach. In the first step, we will generate a saturation mutation library for each critical residue in CDRs. Let's say if you have 30 critical residue in the CDR, we will generate 30 saturation mutation libraries. And we will go through these saturation mutation libraries with SPR affinity ranking, and those beneficial mutation, the one can improve the affinities, will be isolated and built into a secondary library called combinatorial mutagenesis libraries. And after further affinity ranking with the combinatorial mutagenesis libraries, we will end up with the top candidates with the most affinity improvement. Once the sequence is finalized, the next step is the developability assessment. Actually, there is 3 different checkpoints for developability assessment. The first checkpoint is at the time when you do the humanization. Usually we will include immunogenicity predictions, post-translational modification analysis and aggregation analysis along with humanization process. And the second checkpoint is after the sequence engineering. It's a cell-based assays. The primary purpose of the checkpoint 2, the in vitro developability assay, is to evaluate the non-specific bindings, the self-interaction, and the stabilities in plasma, basically in ex vivo stabilities. And in the third checkpoint, which is roughly the time when you do the in vivo animal studies, we will do the manufacturability assessment. The purpose of this step is to evaluate the stabilities of your candidates under stressful conditions like high temperatures, repeated freeze-thaw cycles, and acidic or basic conditions. So that's a general introduction of the overall workflow for single-domain antibody discovery and engineering. And the next, we will share 3 case studies corresponding to each of those platforms I mentioned at the very beginning. So the first case study is the discovery of a GPRC5D leads for T-cell engager bispecific antibody using the alpaca immunized libraries. We already talked about the nature of the target, the GPRC5D, at the first talk, so I will probably skip this information. So similar to that ADC case studies I shared in the first talk, we also use the mRNA to immunize the alpaca to generate the immunized phage libraries. And the library, similar to that hybridoma approach, the library is paired and screening against the engineered overexpression cell line and the myeloma cell lines. After confirmation by flow cytometry bindings and the sequencing, we end up with top 12 candidates. And we select 6 top candidates from this top 12 for T-cell engager bispecific construction and the T-cell activation RGA assays to evaluate the activities of the T-cell activation with these T-cell engager bispecifics. So similar to the previous hybridoma case studies, for this alpaca immunization projects, we also measure the cell titer data using both engineered cell line and the myeloma cancer cell lines. And we also measure the IgGs and the VHH titers. It is a crucial step to measure the VHH titer because most of the camelid animals is able to generate both IgG antibodies and the VHH antibodies simultaneously. And the percentage of VHH antibodies is usually around 30%, but it varies from animals to animals. So that's why it's crucial to make the decision based on the VHH titer, but not solely on the IgG titer, because the VHH is actually the one we need. After generating the libraries, the library will be subject to panning using the antigen cell lines for multiple runs. In this case study, we use the engineered overexpression, high expression cell line for the first round of panning, and use the myeloma low expression cell line for the second round of pannings. After the pannings, we generate 12 top candidates, and we start the more detailed characterizations with these top 12 candidates. The first, again, it's the flow cytometry-based antibody affinities against the myeloma cell lines and the engineered cell lines. In both studies, these top 12 candidates showed equivalent or better cell binding affinities comparing to the clinical benchmark. Based on that affinity data and the sequence cluster analysis, we selected a few top candidates to generate 6 T-cell engager bispecific antibodies with 1 CD3 antibodies. And these 6 bispecific antibodies showed various activation level of the T-cell by these RGA assays. And when we treat these bispecific antibodies with antigen negative cell lines, there is a minimal level of a T-cell activations, confirming that these bispecific antibodies has a minimal non-specific spontaneous activations. It is a pretty crucial indicator for the safety and efficacy of a bispecific CD3-based T-cell engager bispecific antibody. Okay, so that's for the first case study for the immunized libraries. The second case study is the discovery of ROR1 antibody for CAR T cell therapies using the naive libraries. ROR1 is another popular tumor-associated antigen in recent years. It's widely used for T-cell engagers, ADCs, and CAR T-cell therapies. One of the major issues for ROR1 is it has extremely high homology between different species, human, cyno, mouse, and alpacas. So in this particular scenario, if you immunize the animal with ROR1 proteins, most likely you will not generate any immune response because of the immune tolerance issues. So that's why we decided to use the naive library to avoid the immunization issues. So after panning and screening through this naive library, we actually generated more than 400 hits. And based on the sequence cluster information and the relative affinity rankings, we chose 30 of them to move forward for single-chain antibody recombinant production and purification and further characterizations. And based on this data, we selected 8 top candidates which targeted to each of these 3 extracellular domains of ROR1. Next, we take these 8 unique candidates for the CAR construction using this particular structure on the right panels. And we further evaluated the efficacy of the CAR using these 8 different antibodies. The first experiment we did is a luciferase-based killing assays. It's a pretty common assays to measure the efficacy of CAR T cells. From this experiment, we identified top 3 candidates showing equivalent or comparable killing efficacies to the benchmark, the R12. So in addition to the luciferase assays, we also measured the activation of the CAR Ts by the induction level of IL-2 in the middles and interferon gamma on the right panels. Again, the same 3 top candidates gives equivalent or comparable induction level of these cytokines. And luckily, these 3 top candidates accidentally targeted to each of these extracellular domains, so that we chose these 3 top candidates to move forward for humanizations. So each of these candidates were successfully humanized, and the affinity is successfully maintained at 10 to the minus 7 level. Remember, I mentioned that the antibody affinity come out of the naive library is usually worse than the immunized libraries. So that's why we chose one of the candidates to do a further affinity maturation. Affinity is improved from 10 to the minus 7 to 10 to the minus 9. And we use that affinity matured variant to get a further confirmation by the in vitro CAR-based killing and efficacy assays. So that's the second case studies using the naive libraries. The last case study is the discovery of antibodies against an immune checkpoint target using the beacon-based single B-cell approach. As I mentioned at the very beginning, the overall process of the single B-cell based approach is like start with the alpaca immunization, followed by plasma B-cell enrichment, B-cell screening on the chip, followed by pro-speed expression validation and sequencing. The whole process takes about 3 months. Again, during the immunization stage, we monitored the serum titration data again using specific secondary antibodies, either against the total IgG or the IgG2 and 3. The IgG2 and 3 represent the VHH. We also measured the serum titration data against both human and cyno-monkey proteins for the verification of monkey cross-binding affinities. Then we harvested the PBMC from the selected alpacas after 5th and 6th immunizations. And after the enrichment of the plasma B cells, we start the B cell screenings. So for the sample after the 5th immunizations, we identified 156 positive B cells, and 151 of them is successfully PCR'd. The success rate of the PCR is roughly at almost 97%. And for the second batch of the samples after the 6 immunizations, the PCR success rate is also roughly at 95%. And 100% of these PCR fragment is actually confirmed as a VHH, but not IgG. This is a very crucial step because we do not want to end up with a bunch of candidates that is a full-size IgG, but not VHH. After recombinant confirmation, 151 and 179 of the recombinant samples are actually confirmed as a positive binder to the recombinant antigens, and based on the sequence analysis, 58 and 55 of them are the unique sequences. So all of these are pretty crucial indicators we constantly monitor during the B-cell screening stage. And this recovery rate is considered pretty high because the industrial average level of a recovery rate from positive single B cell to confirmed sequence is roughly at 30% to 50%. So after generating this top candidates, we select a few of them for further characterization, starting with binding affinities against recombinant protein using ELISA, or against the overexpression cell lines using the flow cytometries. So as you can see from here, the majority of these candidates showed relatively higher binding affinities in both ELISA and the flow cytometries comparing to these two clinical benchmark. In addition to the antigen bindings, since this is an immune checkpoint target, we also look at the receptor ligand blocking capabilities with these candidates, some of these candidates. And judging by the ELISA-based competition assays, we can conclude that these leads showed a differentiated profile of blockings. Some of them showed really good blocking to the receptor and the ligand binding. Some of them do not. So this further suggests a really good, the diversity is well maintained after the whole screening process. And in the last step, we put all of this sequence together to do a phylogenetic tree analysis, because there are too many sequences, so I just put some of them here. So the yellow highlighted ones are the sequence recovered from the 5th immunizations, and the blue highlighted ones are the sequence recovered from the 6th immunizations. So the sequence recovered from these two independent batch of samples shared a even distribution within different germline of the antibodies, suggesting really good diversities can be recovered from this single B cell based antibody screenings. Okay, so that's all for the talk. Any questions?
질문 있으신 분들은 자리에 손을 들어주시면 저희가 마이크를 건네드리도록 하겠습니다. 네, 혹시 지금 시간 이후라도 질문 있으시면은 저희 스태프분들 통해서 개별적으로 질문 부탁드리겠습니다. 네, 땡큐 자리.
Thank you.